Okay, we are in June 2023. We did 2021. Fine. Paper 2, by the way, is a very, very easy paper, but you need to study really well. And most of the grades, of course, you will gain them from paper 2. So please, before paper 2, study the syllabus really well and solve as much as you can. Now, let's start with this exam. Let me see who I have here. Hello, Mariam. Okay, blood arrives at the heart in the superior vena cava and inferior vena cava. So we will complete here. The blood will then pass into the right atrium. And then when it enters the atrium, 3, continue. This is going to cause atrial what? When the blood enters the atrium.
exam
This is, why do you want to know? This
عشان
is
افتح
June 2023, variant 22.
okay.
So when the blood will enter the right atrium, and then the atrium will do what? Both atria, they will what?
Contract.
Yes, when they contract, this is called atrial.
Systole.
Yes, this is called atrial systole. Now, while the atria are contracting, this is the cardiac cycle. We have 3 stages in the cardiac cycle: atrial systole. While the atria are contracting, what happens to the ventricles? It will be ventricular.
It will relax.
Yes, so we will not write ventricular relax. What is it called?
Diastole.
Yes. Yes, Noor? Okay,
Yes.
Kareem, you will not say ventricular relax, it's ventricular diastole. This is the typical cardiac cycle. We have atrial systole, followed by ventricular systole, followed by the relaxation of the whole heart. Followed by what then? Wake up with me. Wake up with me out of you're sleeping.
Diastole. Systole.
Yes, it will be continued by the diastole. Both the atria and the ventricles, they will relax. So first we have both atria, they are contracting. And while the atria are contracting, the ventricles, they're relaxed because when the atrials contract, the pressure inside the atria will increase and the blood will be pushed into the ventricles. And then the ventricles will contract. And while the ventricles are contracting, the atria is relaxing. And then the blood will go out of the ventricles and then the whole heart will relax in diastole. Give me a second. We said this is June 23.
Yes.
Okay. Fine. And then blood is forced into the right ventricle. And then we just said it. What happens to the atria and what happens to the ventricle? So the atrial, what do we write here?
Diastole.
Atrial diastole and ventricular systole. And then blood leaves the right ventricle to enter the pulmonary arteries. And both here, atrial and ventricular, what? Both of them will relax. So what do we say here?
Diastole.
Diastole.
Yes, atrial diastole and ventricular diastole. Very easy questions. With reference to figure 1.1, explain why it is important for the control of the cardiac cycle that there is a delay, a short delay, at the atrioventricular node after impulses have been sent out by the sinoatrial node. So what is the importance of the delay? Again, it's a direct question. Why is this delay important?
Prevent the
allow
atrial.
the
Okay, tell me. Prevent what? Mariam said prevent something.
Prevent atria and ventricles contracting together.
Very good. So we'll prevent them contracting together. So in which order do they contract and why?
Atrias contract first.
Very good. The
Then
atria, they will contract first. Then
the atrioventricular node will cause a delay of the
very good.
wave of excitation.
Yes, there's a delay. And then what will, after this delay, What will contract after the atria?
The wave of excitation passes through the bundle of his supporting tissues to go to the apex of the heart, and then the ventricles contract together.
Correct. This is more than needed. Now, give me just one reason why the atria, they will contract before the ventricles. Why does the ventricles and the atria not contract together?
To make sure that the ventricles are filled and the atrias are empty.
Very good. So that the atria are emptied and the ventricles will be filled with blood, because if they both contract together, then the ventricles will not be filled with blood. And to prevent, if you want to, if this is out of 3 marks, for example, to prevent atrial fibrillation or to prevent fibrillation in general. This is a very also direct question. Any questions? Stop me. Now, of course, you will read the whole question, okay? Changes in blood pressure occur in the heart during the cycle. These changes cause the opening and the closure of the bicuspid and the tricuspid or the atrioventricular valves, and the aortic and the pulmonary or the semilunar valves. Explain how blood pressure changes, causing the opening of the tricuspid valve, causing the opening of the pulmonary valve, help the flow of blood through the heart. So this is a 3-mark question and it's very organized. First, we want to explain how the blood pressure will change in the first option, Kareem. When there is opening of the tricuspid valve, first, Kareem reminds me, where is the location of the tricuspid valve? This is my heart, divided into 4 chambers. And this is the left side, and this is the right side. Where is the tricuspid valve? This is O level. Is it on the left or the right side of the heart? Kareem.
Right.
Miss, can you hear me?
Yes, I can hear
Right,
you, my dear.
right.
Of course, it's the right. So the tricuspid valve is on the right side of the heart. Where is it exactly in the right side of the heart, Kareem?
In between.
Between the right atrium and the right ventricle. Yes, so the tricuspid valve is here. Now we know that when valves open, they are like doors. There is an increase in the pressure behind the valve or in front of the valve, because they specifically want pressure changes. So this valve will open. There will be, for this valve to open, there will be high pressure behind the valve or in front of the valve.
In front.
Excuse me?
In front.
No, the valve is like a door. It's like a door. So I want high pressure behind the valve so that this door will open, and low pressure in front of the valve. We even took this in O level. Any valve that will open, the valve is like a door like this. We want this door to open, Kareem. So there has to be high pressure behind the valve and low pressure in front of the valve. Are you with me? Okay, Kareem?
Yes.
So explain the pressure changes that will cause the opening of this tricuspid valve. The tricuspid valve is here. This is the tricuspid valve. You gave me the location. I want this valve to open. So the pressure will be behind the valve and low in front of the valve. So the pressure will be higher where? What is behind the valve?
Ventricle.
Habibi, the tricuspid valve is here. It's here. I want it to open. It will open like this. Blood is flowing from the vena cava into the right atrium and then the right ventricle. So the blood will move in this direction. Okay, I want the valve to open. So any valve opens when there is high pressure behind it. So behind the valve, what is located? So there will be high pressure where? Noor, can you help him?
high pressure في الاتريوم.
Yes, in which atrium?
right atrium.
Yes, and low pressure where?
Right ventricle.
Very good. That's it. That's it. So answering how the blood pressure changes that will cause the opening of the tricuspid valve. You can say that the tricuspid valve is found on the right side of the heart, between the right atrium and right ventricle. It's an atrioventricular valve. This is O level. And for this valve to open, there will be high pressure behind it. So there will be high pressure in the right atrium and low pressure in the right ventricle. Finished. That's it. Easy and clear. Okay. Now the second point here, the opening of the pulmonary valve. Mariam, where is the pulmonary valve located?
At the end of the pulmonary artery.
Very good. Is it an atrioventricular valve or a semilunar valve?
Semilunar valve.
Correct. So in points, clear. So this valve is located below the pulmonary artery. It is a semilunar valve. Now, for any valve to open, where will the high pressure be behind the valve? Then the high pressure will be where?
The ventricle.
Yes, which ventricle? Be
The right
specific.
ventricle.
Very good. So high pressure in the right ventricle and low pressure where? What is in
In the
front of this valve? In the
pulmonary artery.
pulmonary artery itself. That's it. That's it. And then the last point, they want the last point. How do the valves help in the flow of blood through the heart? What is the function of the valve anyway?
Prevents the
Prevents
backflow.
That's it. Prevents the backflow of blood. So this is a very easy question. Very easy question. This can be an O-level question. It depends on how well you know the anatomy of the heart and the mechanism how valves open and close. We took this also in year 10, that any valve, any type of valve, whether the semilunar or the atrioventricular valve, it will open when there is high pressure behind it and low pressure in front of it because it's like a revolving door. High pressure will make it open if the pressure is behind it. Okay. If there is high pressure in front of the valve, this valve will close. So we have a valve. This valve will open if the blood is traveling in this direction. The valve will open if there is high pressure behind it and low pressure in front of it. Now, when do the valves close? They close when there is high pressure in front, the opposite. So they are asking you a clear question. Explain how the blood pressure changes when this valve opens, when this valve's open, and what is the function of the valves. So you need to know the location of the valve and mention its location and look for the structure behind it. It will be of high pressure. And the structure in front of it, it will be of low pressure. Clear? Is this question now understood? Kareem?
Yes.
Okay? Because normally anything moves from high pressure to low pressure. Think about it. In blood, blood always moves from high pressure to low pressure. In plants, water always moves from high hydrostatic pressure to low hydrostatic pressure. Always the movement of fluids is by mass flow, and it's always from high pressure to low pressure. Put this in mind. Okay? So we're done with the first question. So this question is actually very easy, but again, they misguide you and they want to confuse you. But the question can be a direct question. How do the valves open? Describe the mechanism of the opening and the closure of the valves. And what is the function of the valves? That's it. Okay. So where are we? We are in question 2. Of course, this is the end of a root tip and they are showing you dividing cells. So this is mitosis. Describe 3 differences visible in figure 2.1 between the root apical meristem and the root capsule. So this is it has nothing to do with how much you studied. You are going to look at these cells and compare between these cells and these cells. Can you give me 3 visible differences? What can you see between those cells? I'll try to narrow it down. Between those cells and those cells, there's a very obvious difference.
More nucleus visible في الروت apical.
Yeah, the root apical, the nucleus is what's visible.
More
Actually,
number.
anything you write, they are smaller. These are smaller and they are more in number. I can see that. And they are maybe longer or stretched out. They're longer or
less
these
less
are larger. Huh?
less cytoplasm visible في الروت apical.
Yes, you can see a bigger nucleus. Okay, bigger nucleus. And less cytoplasm. Okay, fine. And what will you say about those root capsules?
They are larger.
They are larger and they are generally less in number. And what else can you say?
Doctor,
Mm,
some of the cells in the root apical, some of them show the steps of mitosis, while the root
yes, of
none of them
course.
show mitosis.
Yes. Actually, because the meristem, these are the dividing cells, so they obviously show that they are in different stages of mitosis. Of course, this can be another visible difference. Anything that you can see. And in the root capsules, they look more stable and they don't show different steps of mitosis. So this is a very easy question. We will not waste time on it. Just write any differences that are visible. With reference to the cohesive and adhesive properties of water, suggest and explain how the formation of small clumps of soil helps to maintain the soil water around the root tip. Okay, again, it's a very direct question that is basically asking you what is cohesion and what is adhesion in water, and describe it in detail. And you can even draw in this question if you want. And how can this, or suggest, how does this explain the formation of small clumps of soil around the root tip? So you have a root tip and you have small clumps of soil here. So first, what is cohesion in water? What will you say? We took it in biological molecules.
Cohesion is when hydrogen bonds are present between each between water molecules.
Very good. Cohesion first, the definition. This is the attraction between similar molecules or different molecules.
Same molecules, different
Yes,
charges.
yes. Attraction between similar, so you can define it, similar molecules. In this case, the molecules that we are talking about is water. Now, the hydrogen bond. Perfect. So this hydrogen bond happens between what and what exactly? We have a
Hydrogen and
dipole.
oxygen.
Huh,
Hydrogen
very
are
good.
is electro positive,
Mm,
oxygen electronegative. So
very
they
good.
bond together.
Very good. Okay, you can actually make this nice drawing or describe it just like Noor did. The oxygen is slightly electronegative and the hydrogen is electropositive because we have sharing of electrons and this sharing is equal or unequal, Noor.
Uh, Equal.
No, Habibi. If it was equal, it will
Emo-
not have an overall charge. It will be neutral because there is unequal sharing of electrons and there is more attraction of electrons around the oxygen. That's why the oxygen, it has a slightly negative charge. And the hydrogen has a slightly positive charge. This is called electronegative and electropositive. Now, the hydrogen bond is between two molecules of water. So I'm going to draw another molecule of water here. Okay, with an electronegative charge on the oxygen and electropositive charge on the hydrogen. And how do I draw the hydrogen bond? Let me use a different color. Let's say green. And the hydrogen bond is represented like this. Dashes between the electropositive hydrogen and the electronegative oxygen. And you can label it. This will be a very nice answer. This is the hydrogen bond. So the cohesion, what is it? It is attraction between similar molecules. In this case, we're talking about water. And the cohesion is the result of the hydrogen bonds between the electropositive hydrogen and the electronegative oxygen in a water molecule. Easy? Exactly what we took in biological molecules. Now, discuss adhesion. What is adhesion? Hmm,
Forces of attraction between different molecules.
exactly. And in this case, we're talking about the water and the soil. The water and the soil are attracted to each other also by hydrogen bonds. And you don't need to do any drawing in this case. Now, so here we discussed what is cohesion and we mentioned it in detail and we drew it and we gave them everything they want and more. And we talked about adhesion. Now, suggest and explain how the formation of small clumps of soil helps to maintain the soil water around the root tip. A critical thinking question. I have here clumps of soil. How does this help maintain the water around the roots? It's very easy. These clumps of soil, what do they trap?
What?
Yes, they attract water. So the water will always remain around the root tip. Okay? So we talked about adhesion density, attraction between different water molecules. In this case, it will be the water and the soil. Okay, easy. I don't even need to look at the mark scheme. So state the term used to describe enzymes that act outside the cells. Very easy. What is the name? Where's Kareem? Is he here or he left?
I'm here.
Oh, I have a lot of people here. Now Farouk and I have Adham. Okay, Adham, what is the name of the enzyme that acts outside the cells?
Extracellular enzyme.
Very good. Extracellular. Now, the nice thing about enzymes, about exams, is that they make you revise different parts of the syllabus. What is meant by a polysaccharide? Tell me, Adham. Polysaccharide. Biological very easy definition. Poly. Anything poly. Mm. Polysaccharide. A polymer. A polymer is what? Example, polysaccharide.
It's
It's
made
made
of
up
many
of what?
monomers.
Very good. It's made up of many monomers. What are the monomers?
Saccharides, yes.
Huh? Yes, monosaccharide. Maybe I did not hear
Monosaccharides,
you.
yes.
Many monosaccharides joined together. These are 2 marks. These monosaccharides in the case of a saccharide. Okay? Saccharide is a what? It's a carbohydrate or a protein or a fat. How are they held together? Yes, it's a carbohydrate. Very good. And what is the bond in carbohydrate like starch? Very good. Glycosidic bonds. Say it. Glycosidic. And right here, acidic bonds. Okay, you can give examples, but it's only 2 marks. This is enough. So many repeating subunits, many monomers bonded together, many monosaccharides. It's a carbohydrate. It's made up. It's joined by glycosidic bonds. Okay. Draw the general structure of an amino acids and the space provided, and use this drawing to explain how it is possible to have many different amino acids. Now, this exam is mainly biological molecules. What is the structure of the amino acid? You have to know it. Help me. What are the 2 functional groups and why are different amino acids different?
A carboxyl group first.
I have, yes, a central carbon bonded to a carboxyl group. Okay, from one side. And what is the bond to the other side?
Amine group.
Amine group, NH2. Very good. And I'm not finished. This carbon can form 4 covalent bonds. So it is bonded here to what?
To an R group.
Okay, an R group, yes, I will not tell you it's wrong. And this is the reason why every amino acid is different. This R group is called the variable group. Yes, this is the reason why different amino acids are different. And it can be hydrophilic, hydrophobic, charged, whatever. And what is here?
Hydrogen.
Hydrogen. Very good. So this is the basic amino acid structure. And you can describe it here, but they just said draw and label. Okay, so you can label this is the amine group, this is the carboxylic acid group or the carboxyl group or the COOH. And this is the hydrogen, and this is the variable group or the R group that is different in every amino acid. So just to explain how the presence of mineral ions in the root hair cell can increase the absorption of water by the root hair cell. Again, this is an O-level question, very easy. Why mineral ions or salts inside the root hair cell? This is my root hair cell. It is very concentrated. It has a lot of mineral ions. How does this help the root hair cell absorb water? Who did not speak? Where's Kareem? Come on. Yes, my dear. Tell me, why a concentrated root hair cell that is filled with mineral ions helps in the absorption of water? How does water move? Why does it move? Discuss.
Okay, I'm 1 minute. Water potential lower in the cell.
Excellent. Now this cell will be concentrated. It will be filled with salts. So it will have a low water potential, just like Kareem said. And outside, it will have high water potential or low water potential. Outside the cell, there will be lots of water or less water. Yes, correct. So outside, you will have high water potential. And this will help in the movement of water by what?
From
Hmm,
osmosis.
By,
Osmosis.
very good, by osmosis. You can define it if you want, the movement of water down a concentration gradient. This is important across a what? Semi-permeable membrane, because water always moves from high concentration gradient to low concentration gradient. And the different or the higher concentration gradient, the faster the osmosis. I think you know this all from O level. All this is O level, but they just don't want to admit. Okay? Now outline the features of non-self antigens. What are the features of the non-self antigen? What are they made up from? What do they do? Hmm? Who did not speak? Where are you, Mariam? What is a non-self antigen?
It's a foreign antigen that doesn't belong to
Very
the
good. It's foreign. In other words, it does not belong to the body. Where is it located exactly on the cell? What is it made up of? What does it do?
It's present
Protein.
on the surface of the
Usually it is present on the surface. Yes, it's a biological or a molecular marker. It marks the cell. Every living cell has an antigen. This antigen can be self or non-self. The non-self is the foreign. What is it made up of, Mariam, most of the time?
proteins.
Very good, proteins. If it is attached to a carbohydrate, it can also be called glycoprotein. Good. We took in cell membranes that glycoproteins can be antigens. Now, what does it do?
It
What's the
identifies the cell. It has a specific shape.
Yes, it does have a specific shape. Good. All this is correct. Now, what does it do to your immune system?
It stimulates the immune system.
Very good. It stimulates the immune system and everything else that goes with it. Now, if you notice, stimulating the immune system and the story that comes after the stimulation is in every paper 2 exam. Every paper 2 exam, they either have to ask you directly or indirectly about what are the sequence of events that happen in your body after immune stimulation. Okay, so write it everywhere, anywhere you are, like you feel you need to write it, even in short points, write it. Like in this question, that same question can be 3 marks or 5 marks. If it is out of 5 marks, then you will mention that the function of the non-self antigen or its significance is that it will activate the immune system and outline what happens after that. Immune system is activated means I have inflammation, means I have increased blood flow, means I have phagocytosis. Phagocytosis means I have antigen presentation because phagocytosis includes the neutrophils and the macrophages. I think you remember this very well. Antigen presentation means I'm going to activate the T help. I'm going to release cytokine. After the cytokines are released, this is going to activate all the lymphocytes and more macrophages. The lymphocytes are the B lymphocytes or the T lymphocytes. B lymphocytes are activated or T lymphocytes are activated, they go through clonal selection, clonal expansion, and then differentiation. When the B cells differentiate, they give me plasma cells. When the T cells differentiate, they either give me T helper or T cytotoxic or T killer. Anytime the immune system is activated, this sequence of events has to happen. This is primary immune response. Are you with me?
Yes.
Okay, so this question is out of 3 marks. Maybe it does not need all that, but if you write them in points, you will not lose anything. I always advise you to write everything because you don't really know what they want. So the features of non-self antigens, yes, they are foreign usually on the surface of the cell. They are molecular markers, usually made up of proteins. Sometimes they're made up of glycoproteins. And why are they very significant? Because they activate the immune system. Once you mention that the immune system is activated, either outline what happens next in detail or in summary. It depends on the question. In this case, you will outline what will happen in the immune system when it is activated in summary, not in detail. Clear? Are you with me? I want to exploit the chance and revise it again for the 10th time because steps or activation of the immune system, it shows up in every exam. Every exam. I'll do it very, very quickly so that hopefully it sticks. Okay, very, very quickly. I will not waste your time. I have immune system activated. Why is it activated? Because I have a foreign antigen. Always I have a foreign antigen, bacteria, virus, whatever. And then this is followed by inflammation. Inflammation means more blood supply. Capillaries will become more leaky. What is the first line of defense? Who will come with the increase in the blood flow? The phago? Huh?
The phagocytes.
Very good, the phagocytes. We have two types of phagocytes, the neutrophils and the macrophages. The macrophages here, they become antigen presenting cells because they do incomplete phagocytosis. Antigen presenting cell asks for help. This is the easiest way to remember it. Antigen presenting cell will present the antigen and it will ask for help. It will ask for help from who? Hmm? Who is going to be activated next?
The B lymphocytes.
Yeah.
The T helper. The T helper releases what?
Cytokines.
Cytokines. These cytokines, they activate the whole immune system. They wake up everyone else. They will activate the lymphocytes and they will activate more angry macrophages. When the lymphocytes are activated, either the B lymphocyte or the T lymphocyte, each one of them is going to work in a different way. When any one of them is activated, also they go through, here you mentioned clonal selection. I will select which B lymphocyte, the one that has a complementary receptor to the antigen, okay? Clonal selection, clonal expansion. What do I see here? After I select which one is complementary, it will divide by mitosis to increase in number, and then it will differentiate. The B lymphocyte differentiate into plasma cells that produce what? Specific antibodies. And some of them remain in the blood and they become memory. And the same thing happens when the T lymphocytes are activated. They go through clonal selection. I will select which one has a complementary receptor. Clonal expansion, so clonal selection. Clonal expansion, whatever I selected will expand and multiply by mitosis. And then I'm going to differentiate. I will either produce T helper, more C killer cells, and in all cases, whatever has been differentiated will remain in the blood. Some will remain in the blood and form memory cells. Don't forget that the T killer do not produce antibodies. The T killer is called T killer because it directly kills the cell. Direct cellular immunity. It goes to the infected cell and it kills it by the release of chemicals known as perforin or hydrogen peroxide that usually perforate the cell wall of bacteria. That's why it's called perforin. Okay? Or free radicals. They release chemicals that kill the cell. Clear? So here we exploited the chance and we revised this part. Enough. Okay, now cells of the immune system. Immunity is a very important lesson. Which one of them will able to go through colonal expansion? Which one will go through colonal expansion? B lymphocyte goes through colonal expansion? Yes or no?
all except the plasma.
صح. Okay, plasma cells produce antibodies. The main role is to secrete cytokines. Which one secretes cytokine? Again, we just revised it.
T helper.
Yeah.
T helper. Which one is present in a primary immune response?
All.
All this is in a primary. The secondary immune response, memory cells, finished. Memory cells, they wake up, they are activated, they divide by mitosis, they differentiate, and they make the secondary response stronger and faster. Okay, enough immunity. The part of the cell cycle that occurs immediately after mitosis, what is it?
Cytokinesis.
Yes, cytokinesis, good point. The part of the cell cycle in which the synthesis phase occurs.
Interface.
Interface, excellent, Karim. The main event that occurs during the synthesis phase.
DNA replication.
Yes, always use semi-conservative DNA replication. Good. Let's finish. Okay, I have a picture here. I think this is what? This is an organism that is, it causes disease, so it's a pathogen. This disease is called the sleeping sickness. Insects known as tsetse flies pass on the organism. So again, it is spread by an insect here, similar to malaria that we studied, from infected people to uninfected people when the male and the female tsetse flies fuse together. This is a transmission electron micrograph of the pathogen. They just said it's a unicellular organism, but you can here identify this is a virus or a bacteria. or a plasmodium, or what type? It's eukaryotic or prokaryotic?
Eukaryotic.
Eukaryotic.
Yes, of course. So it's not a bacteria. Why did you say it's eukaryotic?
عشان
There's
في
a
organelles.
nucleus.
Yes, because there's a nucleus. Very obvious. It's large and there's a nucleus. I can see different membrane-bound organelles. I can see mitochondria. Okay. So with reference to figure whatever, draw an arrow to indicate the location in the cell where ribosomal RNA and proteins are assembled to make ribosomal subunits. Where is the ribosome made?
nucleolus.
Yes, this is the nucleus. Okay. Now, with reference to figure 4.1, explain how the structure labeled X provides evidence that T whatever is motile, able to move. So what is X? Where is X? This is X. Let's zoom in. Why does X indicate that this pathogen can move? Look at the structure. It's close to something that. Why does X indicate that it can move? It looks like a what? It represents a what? Noor.
I don't know.
Bacteria, usually, what do they have on their surface outside like this?
الفلاجيلا اسمها فلاجيلا.
Yes, okay. So it is found on the external surface. It can indicate or act as a flagella. And the flagella, look at the structure. It's made up of what? These flagella or this X is made up of what? Remember the structure of the microtubules? We said it is made up of alpha and beta tubulin proteins that are usually in pairs, and these pairs are called dimers. Again, this is biological molecules. Okay, if you look closely, you can see that it is made up of pairs of dimers. These are the alpha and beta tubulin proteins that are found in microtubules. This is also in biological molecules. We have 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 times 2 tubulin, but this is too detailed. You can just notice or remember the structure of the microtubules from biological molecules, and you can indicate that it has the structure of a microtubule. It is made up of pairs of dimers. These pairs of dimers are alpha and beta tubulin proteins. It is in biological molecules. Do you remember it? Or you don't? Hello? Are you with me?
I don't
You want
remember.
me to open? I don't remember. Let me see. Where am I? Where? Okay, it's gone. Oh, this is in cell structure, not biological molecules. Cell structure. Where is it? Here. Okay, here. These are microtubules. And microtubules, they make the flagella and spindle fibers and cytoskeleton. These are small proteins that are circular like these. They're called alpha and beta tubulin proteins, and they are pairs. They are usually arranged in pairs called dimers. You have this same picture here in the lesson. See, alpha tubulin and beta tubulin, and together they make a dimer subunit. Okay, Noor? This is very similar to what you see here. This. So they don't leave out any detail. They can ask you about anything. So here they're asking you, suggest why X, structure X, makes this pathogen motile. you can say that it's found on the external surface, similar to the flagella. If you notice the structure, it has a similar structure to the microtubules that make up the flagella, which are made up of tubulin proteins that are arranged in pairs, any one of those. Okay, do we look at the mic scheme? This is which number? I think this is number 4. We have 2 more questions to go here. Any two from suggest it has similar structure as a flagellum, presence of microtubules, and even you can count them, 9 plus 2 pairs in an arrangement pattern. It is located on the outer part of the cell, so it will help it move. Circular cross-section suggests a tubular structure, hence it has the same structure as a flagella, any one of those. Clear? Okay, let's continue. With reference to the figure, explain whether this pathogen is an eukaryote or prokaryote. We just answered this question. We said it's an eukaryote. Give me 3 reasons why it is an eukaryote, Noor. Who do I have here?
It
And
has a
that's
nucleus.
not only Noor. It has a nucleus, number 1. Number 2, Adham, why is this a eukaryote, not a prokaryote? A lot of reasons. Not only it has a nucleus. Yes, it has a nucleus. This is one big reason. What else?
Mitochondria.
Contains.
Yes, it has membrane-bound organelles. It has mitochondria. And I can see here, what's this? This one, the one with small dots. Maybe the picture is not very clear. It's extensive, actually. These. That's continuous with the outer membrane of the nucleus. What's this?
A rough endoplasmic reticulum.
Excellent. The rough endoplasmic reticulum. This is definitely not found in a prokaryote. Also, it looks large. It's larger in size than a prokaryote. You can measure here the scale bar and convert it if you have time. But it is definitely larger in size than a prokaryote. It has a nucleus and it has a double membrane, a nuclear membrane. It has a double cell membrane. It has a mitochondria, membrane-bound organelles. It has a rough endoplasmic reticulum. So there are so many reasons why this is definitely not a prokaryote. Now, state the term used to describe an organism that causes disease. Very easy.
Pathogen.
It's a pathogen. Okay. Name one of the species of the plasmodium that causes malaria. Only one.
Falciparum.
Oval.
Falciparum, oval, malaria. Which one? Outline the similarities and differences between the modes of transmission of malaria and sleeping sickness. Yes, I told you it is similar. What are the similarities? You see, 3 marks. Give
Yeah,
me.
They are transmitted by a vector.
Very good. They are both transmitted by a vector, by an insect. What else?
The insect it feeds on the human blood.
Yes, you are correct. They both feed on human blood. Okay, give me differences. Let's go back and read the question. Okay. Is it the same? We said, what was the name of the fly in malaria? It was the Anopheles fly. Here, it's not even a fly. It's a fly, but it's called the tsetse. Okay? No. In malaria, it's a mosquito. This is a fly. So this is the difference. This is called the tsetse fly. And in malaria, it's a mosquito. What other difference? So I have mosquito here. And this is a fly. What other difference? Let's read. I think I underlined something. When male and female tsetse flies feed on the human blood, what does this remind you?
Malaria only female.
Yes, in the malaria, it's only the female. Okay, is this enough? So only female fly, mosquito in malaria. Okay, enough. You will get the grade. If you find any differences, write them down. Okay. Now this is a direct question. The name of the bases and whether it's a purine or a pyrimidine and whether it is present in RNA or DNA or both. We said there is a way to remember them. We have 2 types of bases. What are they? We have the purine and the pyrimidine. The purine, Adham, is the double ring or the single ring structures?
Double ring.
Double ring. Okay. What are they? Give me examples. What are the two
Adenine,
bases that make? Yes, and
adenine and guanine.
very good. The AG. The pyrimidine is definitely then the single ring structure, and the examples are TUC, TUC. Okay. The name of the base. What's A? Now, this is so easy. What's A?
Adenine,
Quickly.
Adenine.
Adenine. Is this purine or pyrimidine?
urine,
Purine. Now, this is so easy. Presence in RNA, DNA, or both?
both.
Both. Quickly. C.
Cytosine,
Cytosine. Purine or pyrimidine?
pyrimidine,
Pyrimidine. Present in
both.
which one? Both. Now, so that I don't waste your time, uracil is the only one that is present in
RNA,
RNA. And what about thiamine?
DNA,
It's in DNA only. Okay, again, it's a nice and a direct question. This is a nice example. Okay, no, it's not very nice. Figure 5.1 shows the molecular structure of abacavir and carbovir triphosphate. Good. Now I have to read this one. Abacavir is an analog drug used in the treatment of some viral diseases. It enters a cell infected by a virus and it is metabolized to the analog carbovir triphosphate. So this enters the body and then it will change to this. Now, this reminds you of what? Of which structure?
Protein.
No, because it has NH2. Okay, but what has a sugar and then a base? No, this is a base. It's a double ring structure. It's a base. This is a base, by the way. And something here. This reminds me of which monomer? Look at this one. This one reminds me of what? You have a ribose sugar, 1, 2, 3, 4, 5, or a pentose sugar, a 5-carbon sugar, and you have 3 phosphate groups, and you have here a 2-ring base.
بس الصراحة it
This,
reminds me of ATP عشان دي
yes, it
triple.
does. Yes, it does remind you of a nucleotide, which is an ATP is a nucleotide, okay, with 3 bases. 3 bases, 3 phosphates, I mean, reminds you of adenosine triphosphate or the activated what? Hmm, activated nucleotides. Remember the nucleotides during DNA replication? They are made up of a deoxyribose and a base and 1 phosphate group. When they are activated, they have 2 extra phosphate groups that are removed by the DNA polymerase. And when the DNA removes them, this releases energy, and this energy is used to create the base pairing and the complementary base pairing and the backbone. Okay, so with reference to figure 5.1, explain whether the carbovir triphosphate will replace a purine or a pyrimidine nucleotide in the elongate. No, I have to read the question. Carbovir triphosphate, this one, okay, this one, can be inserted into an elongating polynucleotide chain instead of a nucleotide. This interferes with the action of the DNA polymerase enzyme during the synthesis of viral DNA. So even if you don't remember what it reminds you of, they will tell you. So they will tell you that this structure, the carbovir triphosphate, it interferes with DNA replication because it is similar to the nucleotide that the DNA is using. So with reference, explain whether the carbovir triphosphate will replace a purine or a pyrimidine nucleotide in the elongating chain. Now, what do you look out here to know if it will replace a purine or a pyrimidine? What do you look like? What do you
The
look
number
at?
of rings,
The number of rings. This is a double ring. So it will
urine,
replace a purine, of course, because a purine is a double ring. Easy. Just read the question carefully. Okay, Kareem, is everything clear? Mariam, everything clear? With reference to Adham, with reference to figure 5.1 and the action of the DNA polymerase, suggest why the conversion of abacavir to, I don't know what, increases the chance of the analog being added to the viral polynucleotide chain. So you need to understand this question. This question is also very easy, but they like to complicate things. In other words, with reference to the figure that you're looking at, and the action of the enzyme that you already know, suggest why Adham, the conversion of abacavir to sarbovir triphosphate increases the chance of the analog being added to the viral polypeptide chain. In other words, Adham, why will we use this instead of this, okay, in the making of DNA? Or why should we use this in the making of the DNA instead of what we should use?
Because it has phosphate.
Because it has phosphate. What do you mean because it has phosphate?
Oh, I'm confused, Assassin. I don't understand.
Yes, the question is confusing. I'll try to simplify it. You know what is the action of the DNA polymerase?
Yes.
Yes, it works in the replication of DNA. What
Yes.
does it do?
Unzips and unwinds the DNA strand.
Very good. And what does it add?
Reactivated the nucleotides.
Exactly. You just said it. So I have here these strands after they have separated, and I'm going to add a complementary base, and I'm going to create the new complementary strand. Now, this was an activated nucleotide. Yes, you just said it. Why was it activated? Because it had two extra what?
Phosphate groups.
Very good. And what did the DNA polymerase do?
Break those bonds.
Yes, it removed the two extra phosphates. And when it did that, this created energy, and this energy was needed or was used to create the new sugar phosphate backbone. Yes, this is the action of the DNA polymerase. Don't you know this from before?
Yes.
Good. Now, they said in the question, use the figure and the action of the DNA polymerase to explain why the DNA polymerase will use this instead of the activated nucleotides. Look at the figure. This is an enzyme here. We're talking about the action of the enzyme. Enzymes react with specific substrates. Why will the enzyme DNA polymerase not react with its substrate, but act with this instead? This and the substrate are what?
Complementary to the
Complementary.
enzyme يعني.
Exactly. Yes, they are similar. So this and the substrate are similar, and they are both complementary to the active site of the enzyme DNA polymerase. How are they similar? Look at the figure. They both have three phosphate groups. They both have a purine. They both are purines. They are both similar, so they will both compete for the DNA polymerase because, just like you said, they are both complementary to the active site. Okay?
Okay.
Good. They are similar, but because they told you, look at the figure, you have to indicate how are they similar. Both of them, the carbovir triphosphate and the activated nucleotide, they both have three phosphates, and they both have purine bases, for example, and they are both complementary to the active site of the enzyme, and they will both compete. When I have two things competing for the same enzyme, then one of them will be a what? When
Inhibitor.
I have the real substrate and I have something else that looks like a substrate and they're both fighting for the enzyme, this other thing will be it starts with an I, inhibitor. Very good. So actually, it's easy, but they like to confuse you again. Just read, when you don't understand a question very well, read it again. Okay? Read it again to understand. Let's look at the mark scheme in case we are making up biology. Okay, this is question 5. I think here. Look for any two from, it's only 2 marks, because the carbovir triphosphate is similar or same as the activated nucleotide. Okay, very good. Okay. And ignored purine. So if we said they're both purines, we will not use a mark. They will ignore it. But we said they're similar. We said they are similar to the activated nucleotide. It has 3 phosphates. We said it. It's activated. We said it. The DNA of the DNA polymerase action. DNA polymerase, we said that it will create the chain or the phosphodiester bond in the growing chain. It will not do its action because it has an inhibitor. Example, so those 2 are enough. Detail of the DNA polymerase, its action. We know its action. It adds the nucleotides to the growing chain. It forms hydrogen bonds with complementary bases, any one of them. The nucleotides are the substrates for the DNA polymerase. We said that. Phosphodiester bonds are made between the new nucleotides, so it will not be able to do it. Not on the any valid point. This is a better fit. We did mention active site and we did mention complementary to the active site. So clear? So this question is just tricky. It's not hard, it's just tricky. Okay. Triple I. Suggest and explain how this also interferes with the action of DNA polymerase and how this may prevent the synthesis of viral DNA. 4 marks. It is actually very similar to what we just said. So, what will you say here, Noor, Mariam, Kareem? What will you say here? How will this thing, interfere with the action of the DNA polymerase? What is this thing? What is this carboxyl triphosphate when it has similar structure to the substrate and it is fighting for the active site? What is it? We just said it, by the way.
Competitive inhibitor.
It's a competitive inhibitor. Good, because it has then what? Okay, 4 marks, Mariam. What will you say?
It has a similar shape to the substrate of the enzyme, therefore it competes with the substrate to bind with the enzyme.
Yes, very good. I want you to write every keyword that crosses your mind because this is 4 marks. You said active site, yes? You said it will bind with the active site, yes? Instead of the inhibitor. What else can you say? What are the functions of the DNA polymerase that it will not be able to do?
It won't be able to form the sugar phosphate backbone.
Yes, no sugar 4 marks. Okay, so don't forget that. No sugar phosphate backbone, no making of phosphodiester bond, no proofreading of complementary base pairing. The DNA polymerase is not responsible for pairing the bases, but it proofreads. So no proofreading. The enzyme will not do its work. Why? Because I have a complementary inhibitor. And discuss the complementary inhibitor. You have a lot of things to say about it. Similar in shape. It competes with the active site. Its presence will reduce the affinity of the enzyme to the substrate. The enzyme will not do its work. Mention a list of its work. No phosphodiester bond, no sugar phosphate backbone, no DNA replication, no proofreading. Okay, that's why it's out of 4 marks. Now, this exam doesn't end, it ends. Okay, name the airways in generation 1 labeled X in figure 6.1 that branch from generation O. Kareem, what branches from the trachea? What is one? Trachea and then what? Again, this is so easy.
Miss, wait a second. Let me see the picture.
This is the last question? The last question. Trachea and then it branches to give you what?
Bronchi.
Bronchi. Very easy. This is Olaf. So I'll write here bronchi. Okay, stay with me. Suggest why airways from generation to generation are not able to carry out gas exchange. Why would a structure in the respiratory system not carry out gas exchange? What are the features of an efficient gas exchange? This is an O-level question.
This
How
can I
does
say?
gas exchange? Yes, yes, yes.
They have alveoli which are thin.
Yes, yes, thin. Then they're thin. So gases will exchange by what?
By
How do gases move across the membrane or across the alveoli membrane?
by diffusion.
Yes, by diffusion. And for efficient diffusion, alveoli, they are thin, just like you said. What other features in an
They're
alveoli?
increase يعني they are a lot, so it increases surface area.
Correct. So we will write the opposite. Think of the efficient features of the alveoli, and we will write the opposite. Why will gas exchange not take place? Because diffusion will not be efficient. Because they will not be thin, not thin. Alveoli are thin for faster diffusion distance. Now, these will be not thin. You told me alveoli has a good blood supply. So you will say that there is no good blood supply. Now, the good blood supply maintains a what is the advantage of a good blood supply?
Um,
To
I mean,
maintain a concentration difference so that I have high oxygen at one end and high CO2 at the other end so that they will diffuse from high to low concentration. Two things will maintain a good concentration difference. The high blood supply and the good ventilation. Don't forget that. If I have any structure that will not provide efficient gas exchange, then the diffusion will be inefficient. So it will not be thin. It will not have a good blood supply. It will not be well ventilated, not well ventilated. The opposite. Okay. It will not be of high surface area. It will be of low surface area. I have many, many millions of alveoli to increase the surface area. The alveoli, they're moist for the dissolving of gases. It will not be moist. It will be dry. Maybe it will have mucus. If you remember bronchioles and alveoli, they don't have mucus. Mucus is only found in the trachea and the bronchi. Bronchioles, they don't have goblet cells. Okay, so the opposite of an efficient gas exchange system. So again, this is O level. Now we have here a graph. Figure 6.2 shows the results of the HP gas MRI study compared with Weibull lung model for generations 0 to 5. The Weibull lung model was used as a standard reference for airway diameters. Now, this sentence is very important. Here there is a bar chart that is showing you, you have to look at the key, the Weibull lung model. Why did I underline this sentence? Because this Weibull lung model is the standard reference for airway diameters. If you look at the graph, you have the airway generation, in other words, the structure of the respiratory system and the diameter. Okay? And you have two things that measure the diameter, something called projection and multi-slice. And we compare it with the Weibull lung model. Why did I say that we will compare it with the Weibull lung model? Because it is said in the question here that the Weibull lung model is used as a reference for the diameter. Okay, now they will ask you questions, of course, related to the bar chart. State the relationship shown in figure 6.2 between the airway diameter and airway generation for the Weibull lung model data. So you will go to the bar chart and you will only look at the Weibull lung model in generation 0 and 1 and 2 and 3. What do you see in the Weibull lung model?
As the generation increases or passes,
Yes.
the diameter decreases.
Very easy. Okay. With reference to the data, also explain whether this MRI could be a useful alternative to HRCT in obtaining airway diameter measurements. Now, don't do like me and go direct to the question. You have to read it. This high resolution, whatever, is an imaging technique that is used to obtain measurements of diameters in people with lung disease. Regular repeated imaging must be avoided because it involves exposure to radiation. Scientists research the use of a different technique that does not involve harmful radiation. Two types of imaging, known as projection and multi-slice, were used to obtain measurements. So I hope you understand the question now. They are measuring the diameter of different structures in the respiratory system. And why are they looking for different methods? Because the traditional method, this high resolution computed tomography, used to produce harmful radiation. That's why they were thinking of other methods, and they were using the Weibull lung model as a reference. So with reference to the data, explain whether this MRI is useful, alternative to the traditional one in obtaining airway diameter. What will you say? The multi-projection and the multi-slice, these are the two things that relate to the, where is the last question? To the HP gas MRI. Is it useful to use them instead of the traditional high resolution computed tomography? Look at the,
No,
why not?
because they have the same data. And they're almost the same.
So if they have the same data and they are the same with the Weibull lung model, which is our reference, this is good or bad? If the diameters are close, this is my reference, always this is my reference. Are they close to the reference or they're far away from the main reference?
Close.
They're close. So this is good or bad?
Good.
Good. Something else that will make them good is that they do not produce harmful radiation. It is already mentioned in the question. Like, why are we trying those two methods? Because they do not produce harmful radiation. And their results are close to the standard. And you can use, this is all what you will use, say, by the way, and you will just use numbers from figure 6.2. For example, in airway generation 4, they are very close to the model, which is called the Weibull lung model, and give numbers, like go and get numbers, actual numbers from the graph. This gives you marks. So you can either say yes or no. If you say no, then you will say because their results are very different from our model, and you will give examples. If you say yes, then you will say that their results are very close to the model, and you will give examples. That's it. Okay? Plus, they do not produce harmful radiation. Are you all with me? Adham and Mariam and Karim, are you with me? Or am I speaking too fast?
I'm with you, Amos.
Okay, this is the answer. Look here. You will either say, yes, it's useful, or no, it's not. And you give your opinion, and your opinion has to be scientific. If you say, yes, it's useful, you will say that the results are close because you will compare it to the standard and the results will be similar or they will be close. And you have to give numbers from the graph. You have to quote and you have to give data to support always. Okay? And you will say that it is not harmful. And this is already mentioned in the question. If you say no, it is not useful, then you have to say that the numbers are not the same. They're not close to the standard. And you have to also give data to support. That's it. So again, I keep on telling you the questions are easy, but because they ask the question in a tricky way, they mislead you, they confuse you. We finished the exam. This is a very easy exam, by the way. Who has any questions? Let's look at the exam in total. Who has any questions? This question is not hard. It's very easy, but the question format is not very nice. Let me see. This question was tricky, by the way, because the structure of the microtubules is something I don't think that you will focus on while you are studying, and this is something that you might forget. So I will highlight this question so that you go back and study the structure of the microtubules from the first lesson. This was a very easy question. This was O-level. Cohesion and adhesion, so easy. This was an easy exam. It wasn't a hard exam. But easy exams, they have a very high curve. That's the problem. This is, believe me, it's an O-level exam. Anyone has any questions?
No.
Okay, I'll meet you. What's today? I'll meet you next Monday for another exam. And along the way, if you have any questions while you're studying, send me. Fine?
Great.
Okay.
Okay, maybe
Thank you.
next week, next Wednesday, we might go to the lab. So I'll tell you before it.
Okay.
Can
Okay,
it be in the
bye-bye.
beginning of the day?
Yes, it can. At 9 a.m., yes, it will be because this is my timing anyway.
Oh, okay.
Okay? Bye.
Okay. Bye.